Good evening, everyone. Today I'll be discussing the contents of week 3 of Basics of Biology and discussing some questions afterwards. Let me just share my screen. Um, okay. So, um, in the previous weeks, we have learned about classification of or different organisms, how they are stratified for uh, to make understanding them easier. We've also learned about evolution or current how the current set of living organisms have evolved from the organisms of the past. In this week's contents, we will be understanding what uh, cells are, what are different kinds of cells that one would encounter if they were going through the whole set. So moving forward, cells in case of higher eukaryotes, multicellular eukaryotes, in case of say vertebrates, there are multiple organs that perform specific functions, such as liver, kidney, heart, etc. All of these organs have specific tissues that only function in that particular organ, and each of these tissues are composed of cells. So essentially cell becomes the structural and functional unit of life. uh, so therefore the cell needs to have all the infrastructure essential for different functions that would be required for that particular tissue. So on the basis of cellular structure, cells can be termed as either prokaryotes or eukaryotes. Prokaryotes are mostly single cells, but there are always exceptions to every case. In certain cases, prokaryotes develop relationships within each other, and they function as a coherent group of organisms. Um but mostly when you encounter prokaryotes, they are single cells. Eukaryotes on the other hand are can be single cells, multicellular, or can have a organ level or a organ system level of organization. So primary differences that one would see within prokaryotes and eukaryotes are, first is the size, where prokaryotes are really small. They are both of these are in micrometer range, but prokaryotes are usually smaller than eukaryotes, and eukaryotes have a more variable size. Coming to genetic material, prokaryotes have circular DNA present in the cytosol as a free material. So if you take a cross section of prokaryotic cell, there is no division in different structures within. There's a clear division in different structures within the prokaryotes. Rather, the DNA, the genetic material of prokaryotic cells are present kind of like a noodle soup, uh, free floating inside the cytosol. Whereas in case of eukaryotes have the please stop me if there are any questions. In case of eukaryotes, the genetic material is encompassed in a double membrane nucleus. It's a double membrane bound nucleus. There are channels that connect the nucleus to the cytosol, but there is no direct connection. Now next, coming to replication. So origin of when you study replication in the later slides, you would come across a term known as origin of replication. This is a point in the double stranded DNA where the replication machinery assembles itself and then starts replicating the strand of DNA. So in case of prokaryotes, the replication happens at a single site, So therefore it has a single origin of replication. Whereas in case of eukaryotes, there are multiple origins of replication throughout the genome. Next, when it comes to the genetic material itself, prokaryotes, probably because of their small size, do not have introns. Now, the complete genome of a eukaryotic organism can be divided into exons and introns. So exons and introns. Exons are the ones uh, which have the genetic information for creating various proteins, for creating messenger RNA, all of that stuff. Whereas introns were initially thought to be useless pieces of DNA present in the organisms. However, recent studies have shown that intronic uh, intronic regions in a genome also have their own functions. They might not synthesize proteins, but they are responsible in synthesizing other stuff which is essential to the cell. But this is something that we will be reading in detail later. But rule of thumb, prokaryotes do not have introns, and therefore their genetic size is also smaller. Eukaryotes have both introns and exons. Exons are the coding exons are termed as the coding regions of the genome, whereas introns are termed as non-coding regions. Next, organelles. Prokaryotes do not have clear division within the organelles. They do not have any membrane-bound organelles, whereas eukaryotes have membrane-bound organelles, and each of the organelle has a specific function. Next comes ribosome. So ribosome is the machinery. Ribosome has protein synthesizing machinery that is essential for every cell. The ribosome itself is a very complex complex, or not, it's not an organelle, but it's a complex structure which has different subunits. In these subunits come together to form a 70S or a 80S ribosome. Prokaryotes usually have 70S, whereas eukaryotes have 80S. This S here is known as sedimentation coefficient. If you were to centrifuge these ribosomes, they will sediment at different regions in a tube, and therefore these terms are there. These 70S and 80S ribosomes can be further broken down, but you can read about this in detail if you want, but not necessary for the current stage. This will come later when you are learning about translation of RNA, where you will see how the ribosome is structured. Next comes transcription and translation. Because prokaryotes do not have a division between the nucleus and the cytosol, the transcription and translation both occur together, whereas in case of eukaryotes, transcription occurs in nucleus, whereas translation in cytosol. In case someone does not know, transcription is the process of copying the information present in DNA form, in the genetic material of the cell, into RNA form, whereas translation processes the information in the RNA into protein form. Okay. So now we know there are prokaryotic cells and eukaryotic cells. How does a prokaryotic cell look like? This has a much simpler organization as compared to eukaryotic cells, because it does not have any partitioning in itself. It's mostly it's nucleus, it's cytoplasm, and it's the cell membrane and cell wall. Let's go through all of these structures one by one. The first structure that you would encounter is outer flagella. It is present in most bacteria, but if a bacteria is not motile, if it does not require movement, if it's a free floating or if it's a sessile bacteria, it might not have a flagella. So the flagellum is attached to the bacterial capsule, especially in a motile bacteria. It provides motion or locomotion to the bacteria and is responsible for chemotaxis of bacteria. What chemotaxis means is that if you had a source of a chemical which of a chemoattractant, a chemical which is beneficial to the bacteria will tend to move towards that chemical. This process is known as chemotaxis, where the the placement of a chemical at a certain point creates a chemical gradient of sorts. It can be glucose, it can be sucrose, it can be anything. This chemical gradient signals the bacteria that it needs to move in that particular direction towards the food source. There are different types of, so chemo means chemical and taxis means movement. Depending on what kind of phenomenon you are looking at, the terminology changes. There is durotaxis, there is phototaxis, where the organism moves towards the light, which is something that you see in some plants. But anyways, I am digressing. But chemotaxis means that the bacteria tries to move towards the chemical chemoattractant. So it can be chemoattractant or chemorepellent. If it's a chemoattractant, it will move towards the source, whereas if it's a chemorepellent, it will try to move away from. If it's something that is harmful, it will try to move away from the source. Is there a question? Okay, no. Okay. It's the flagellum is regulated by motor proteins present inside the cell and the flagellum. It is governed by ATPases present at the bottom of the shaft, and it is made up of a protein named as flagellin. And if the flagellar protein is reduced or deleted in a bacteria, the infectivity and ability of the bacteria will reduce. Okay. Then the bacteria itself has three layers to protect the cells from external damage. The outermost layer is made up of polysaccharides, and it's the thickest layer. It's of very high molecular weight, and it's impermeable to water and other aqueous solvents. This is the layer that immune cells would recognize and mount an immune action against. Next is the cell wall in the bacteria that is responsible for gram staining, which is the basis of classification in bacterial species. Bacterial species can be classified into gram negative or gram positive bacteria. Okay. Now, gram positive bacteria have there is slight difference in the there is a difference in the structure of how the cell wall is. Based on that, the staining of the reactivity of the bacteria towards gram stain differs. The peptidoglycan layer is thick in gram positive bacteria and thin in gram negative bacteria. This peptidoglycan, so peptido means peptides, and glycan is usually usually pertains to polysaccharides. So in layman terms, you can think of it as a mixture of proteins and sugars, or amino acids and sugars. This peptidoglycan is in case of bacteria, peptidoglycan is a polymer of N-acetyl glucosamine and N-acetyl muramic glucosamine. And so the way they are linked is a beta 1,4 linkage, and there is a whole biochemistry behind this. The sugar polymer is attached to the peptide chain, which is composed of amino acids. By the amino acids that make the peptide part of the peptidoglycan are mostly L-alanine, D-glutamine, L-lysine, and D-alanine. And they form kind of like a meshwork in case of gram positive cells. And if in many cases, antibiotics such as penicillin would attack the peptidoglycan synthesis machinery present in the bacteria, whereas things like lysozyme will degrade the peptidoglycan layer by cleaving the bond, the beta 1,4 linkage bond, this specific bond, which is known as glycosidic bond, that connects energy to NAM to degrade the polymer and make the bacteria susceptible to further treatments. Okay. Um, One more difference that is present in case of gram positive versus gram negative bacteria is the presence or absence of lipoic acid. Lipoic acid is present in gram positive bacteria, while polysaccharide or LPS is present in gram negative bacteria. And both of these have their own roles in antigenic determinant. They, as an antigenic determinant, these are recognized by the immune cells, and the immune cells mount a response against it. And in a lot of experiments, what people do is they inject different animals like mouse with lipopolysaccharides to mount an immune reaction, where they're just concerned about an immune response. They can one of the controls is injecting lipopolysaccharides. Now, what is gram staining? It the there are It's a very long process, but during the staining, the bacteria is stained with two dyes, crystal violet and saffirin, and they are washed with non-polar solvents such as alcohol, acetone, etc. And so in the end, what happens is, because of the thickness and because of the organization of the gram negative and gram positive bacterial cells, gram negative bacterial cells leave with a blue stain due to the thin peptidoglycan layer, whereas gram positive bacteria retains both crystal violet and saffirin stains and appears as pink. So if you were to stain a mixture of gram positive and gram negative bacteria, you will see some some showing blue color and some showing pink color. The other structures that are present in prokaryotic cells is the cytosol and other organelles, which won't be membrane bound, but things such as ribosomes can be differentiated. So they don't contain membrane bound organelles, but the organelles present in cytosol such as ribosomes, genetic material, and in case of bacteria, electron transport chain complexes are found embedded in the plasma membrane. Next comes the genetic material or the chromosomal DNA. As I mentioned earlier, bacterial chromosome is usually in form of circular DNA. It has elements for replication, transcription, and translation, and it follows a rolling circle mode of DNA replication because it has only one origin of replication. Some bacteria may have extra chromosomal DNA known as plasmids. These plasmids can be transferred from one bacteria to another bacteria, and this process is known as horizontal gene transfer. Because both of these bacteria are in the same generation, any transfer that happens between these two is known as horizontal gene transfer. And usually, if one bacteria in a population of bacteria has a has a plasmid which has a resistance gene, it can transfer that plasmid. It can make copies of that plasmid and transfer it to other individuals in that population, and this is how bacterial resistance is built. Okay. In the bacterial plasmid can be found in multiple forms. It can be nicked, or in an open circular DNA format. It can be supercoiled, or it can be relaxed covalently closed circular DNA. Okay. A bacterial plasmid can have multiple structures embedded into it. One is the origin of replication. As I said, this is where the replication starts. It can have an antibiotic resistance gene, or another gene that is responsible for it to resist rejection from the host. So it will usually have some gene that will be beneficial to the host, because having a plasmid also comes with some costs. Apart from both of these things, there are multiple restriction enzymes that are present in a bacteria. These might not be that much helpful to the bacteria, but in our case it helps us to clone different genes into the plasmid and do our experiments. Okay, coming to isolation of plasmids. So you have a bacteria which has a plasmid which you want to get, which you want to extract. What you do is you amplify the bacteria, you grow them in bigger cultures, you then centrifuge it at a particular speed, and then the bacteria will have both chromosomal DNA and plasmid DNA, or the chromosomal DNA is known as genetic DNA, whereas the plasmid DNA can be known as extra chromosomal DNA. Then comes the lysis step. So before lysis, the bacterial pellet is resuspended in solution. So there are three solutions that come: solution 1, solution 2, and solution 3. First, it's resuspended in solution 1, which has glucose, trisacil, and EDTA. Then this creates a isotonic solution of sorts, where the bacterial pellet is present in suspension. What some people do is they add RNases at this step, in the first step. Here with the solution 1, they also add RNases. The RNase will degrade any RNA present in the bacterial cell, so that you get a more purer population, because when you are precipitating out the plasmid, you are not only precipitating out the plasmid, you are precipitating any nucleic acids that is present. So if you have if you have RNA as well, you will get an RNA contamination in your prep. Then is the lysis step. So this particular process is known as alkaline lysis. So it uses NaOH and SDS to lyse the cell and denature the DNA. In this step, in the lysis step, both plasmid and chromosomal DNA will get denatured. Next is the renaturation step, which is done with potassium acetate and glacial acetic acid using solution 3. In this step, the small DNA or plasmid renatures back quickly because of its smaller size, whereas chromosomal DNA remains in a denatured form. So in step 3, you will see a cloudy white colored precipitate forming, which when you centrifuge, the supernatant will only have the plasmid DNA and protein. To separate out, so you have deleted the chromosomal DNA, now you want to separate out the plasmid from the protein. To separate out the plasmid from the protein, you can do a chloroform phenol isoamyl alcohol step, which creates a biphasic layer of sorts. So this will be the aqueous layer, this is going to be the organic layer. All the proteins will go into the organic layer, whereas the aqueous layer will have your plasmid of interest. Then following this, it can be precipitated with 100% alcohol. This alcohol can be isopropanol or ethanol, either of those work. And then with a few 70% ethanol washes, then you can dry the pellet. Sorry. Then you can dry the pellet and mix it in tris EDTA or in water. Instead of using all of this, what some people do is they use columns which are silica based columns. The columns are designed in such a way that they'll attract nucleotides. So in that case everything up until step 3 is the same. After step 3, you centrifuge it. Instead of using chloroform phenol here, what you do is you pass the supernatant through the column. The column then has the column bed has the plasmid DNA, whereas the flow through will have the protein and all of the extra stuff. then you can elute out the protein from the column, elute out the plasmid from the column. So this is how plasmids will look. uh, the first band here is, it's the covalently closed circles. The second one is open circle, and the third one here is supercoiled. Because supercoiled is very compact, it can travel further in the gel. If you are not using RNases, then you will also see a RNA band here. Okay. Next comes eukaryotic cells. Now, within eukaryotic cells also there is a difference in structure in plant and animal cells, but mostly the major structures are the same and the major functions that would happen are the same. Coming to the differences within plant and animal cells, first one is the presence or absence of cell wall. Plant cell has a valid cell wall, whereas animal cells don't. uh, Plant cells are usually larger in size as compared to animal cells. Chlorophyll is present in plant cells, whereas absent in animal cells, except in some cases such as the euglena, which has a uh, dual trophism. It can either be autotrophic or heterotrophic. Vacuole in case of plant cells is large and central here, whereas in case of animal cells there are multiple vacuoles, but all of these vacuoles are small and they're spread throughout the cytoplasm. Mitochondria is few in plant cells, whereas it's more in animal cells, because uh, the animal cells need to synthesize more energy. Lysosomes mostly absent in plant cells, whereas they are present in animal cells. Glyoxysomes are present in plant cells, whereas absent in animal cells. Cytokinesis or cell division happens by plate method in plant cells and by constriction in animal cells. We'll come to this in later lectures. So the first major portion of plant or animal cell is the cytosol, which is the liquidy part of the cell. It has water, salt, macromolecules, everything free floating. It also is a medium where all of the organelles are present. It has an array of microtubule fiber running through it to give the cell some structure, and these microtubule arrays also act as transport machineries, as highways for different cargoes to transfer within the cell. So cytosol is known to exhibit sol-gel transition, where if needed it can be more gel-like, but this process requires multiple biochemical and cellular processes, multiple changes to occur within the cytosol. There is no well-defined function, but it serves as a medium to exchange material between organelles. The organelles are present within the cytosol itself. It also plays several cellular roles such as signal transduction and translation. Cytoplasm is cytosol plus organelles, whereas cytosol is just the liquid part. Coming next to nucleus, which contains the genetic material. It is the central processing unit of the cell. The inside of the nucleus is filled with a liquid called as nucleoplasm, like cytosol. In case of nucleus, it's nucleoplasm. This liquid has nucleotides, enzymes to perform replication, transcription, DNA damage, etc. It has the DNA present in a complex fashion involving multiple proteins, histones to pack into nuclear bodies or chromosomes. So within a nucleus, the chromatin can be present within a eukaryotic nucleus. This division is not present in prokaryotic nucleus. Within a eukaryotic nucleus, the nucleus can be differentiated into euchromatin or heterochromatin. Euchromatin is usually the transcriptionally active part of the cell, whereas heterochromatin has genes which are not essential for a particular cell type. So the DNA, so in case, I don't think there is a structure, but in case of So there are these histone structures around which the DNA will wrap itself around these histones. The placement between the histones will dictate if the chromatin is heterochromatin or euchromatin. If it's very densely packed, it will become heterochromatin. If it's very loosely packed, it will become euchromatin. Earlier it was thought that this euchromatin heterochromatin condition of different chromosomes is consistent the uh, throughout the lifetime of a cell. However, recent studies have shown that this is a much more dynamic process with exchanges happening between the euchromatin and heterochromatin region. A region which is heterochromatin in nature might not stay that way if there is a requirement for certain genes. Okay. Um, Next, coming to the covering of the nucleus, this particular thing is known as nuclear envelope. It's a bilayer. It has two layers of membrane. The outer membrane is continuous with the rough endoplasmic reticulum, which is something that will come in a few slides, and has ribosome attached to it. The space between the two membranes is known as perinuclear space. Here, intermembrane space or perinuclear space. The nuclear envelope has nuclear pore, which facilitate the transfer of synthesized RNA from the nucleus to outside of the cell, and any proteins that are required inside the nucleus from the outside of the cell, from the outside of the nucleus. The nuclear pore itself is 100 nanometer in diameter, consists of several proteins. It's a very complex structure. It might be an active or a passive process, depends on the concentration gradient. Okay, so transport between one side of any envelope to the other side can be of two types, active or passive. If it's an active process, it means that the cell is utilizing energy to facilitate that transfer. If it's a passive process, that implies that the cell is not using energy, rather it's using some sort of a gradient to facilitate the transfer. Usually in a passive transport, what happens is one side of the membrane has a lower concentration, whereas the other side has a higher concentration. Usually the transfer happens from a from a higher to a lower concentration. This gradient might be opposite in case of active transport. Because it's using energy, it has the ability to transfer things from a low concentration gradient to a high concentration gradient, therefore concentrating whatever uh, reagent or whatever chemical is there on one side. Okay, so these nuclear pores can act as gateway for transfer of materials between nucleus and cytosol. RNA formed after transcription from the DNA within the nucleus move out from the nucleus into the cytosol. Proteins that are formed in the cytosol cross the nuclear pore to initiate replication, transcription, and other processes. Even histones are synthesized outside and then brought in. Next comes mitochondria, which is known as the powerhouse of the cell. This is responsible for generation of ATP, which is the energy currency of the cell, like we have rupees or dollars or euros, whatever. In case of cells, it's ATP. This is essential for running various cellular activities. Mitochondria also is a double layered membrane bound organelle with different structural properties. The outer membrane is smooth and cover the complete organelle with large number of integral proteins known as porins. This porin allows for free movement of molecules less than 500 Daltons or 5 kDa within and outside of the mitochondria. Larger molecules move through the mitochondria through transporters involving signal peptides known as mitochondrial targeting sequence. When the if any protein is traveling to the mitochondria, if this is the protein sequence, there will be a mitochondrial targeting sequence. The mitochondrial targeting sequence ensures that the protein reaches the mitochondria and not some other organelle such as Golgi or nucleus. The inner membrane is folded to form cristae, and this cristae occupies major area in the membrane surface and houses mechanisms for anaerobic oxidation and electron transport chain to produce ATP. The reason this is folded is to increase the surface area. The mitochondria itself is very tiny. Because the inner membrane is folded into cristae, it ensures that there is a larger surface for all of these machinery to be present. Okay. Mitochondria can be divided into two parts, the inner and outer membrane, and the space in between, which is known as the intermembrane space here. Protein present in the intermembrane space have roles in programmed cell death or apoptosis. Matrix is the liquid part present in the innermost mitochondria. It contains ribosome, DNA, RNA, enzymes, runs on Krebs cycle and other proteins. So this portion is the matrix. Mitochondrial DNA is circular. It can synthesize its own RNA. It can synthesize its own proteins. There are multiple differences that are there in between mitochondrial DNA and the DNA present in the nucleus. A lot of recent studies have shown that mitochondrial DNA the mitochondria might have been a prokaryotic organism which was encapsulated into the eukaryotic organism, and it still has the remnants in form of circular DNA. But this is just there are some experiments that have validated this, but at this point some of this is still a hypothesis. Okay. Next, coming to electron transport chain. This electron transport chain has multiple complexes, complex 1 to complex 5. These complexes are multi-protein units. Two or more proteins have come together to form one complex. These are present in the inner membrane of the mitochondria, and during reactions such as glycolysis, Krebs cycle, all of these processes produce NADH2 and FADH2, both of these chemicals. These the electron transport chain processes these NADH2 and FADH2 molecules and generates proton gradient across the membrane. So the intermembrane space and matrix have a difference in the proton gradient, in the proton concentration, due to which the proton is expelled into the intermembrane space and returned back to the matrix to complex 5. The complex 5 is known as ATP synthase, as it generates ATP because of this flux of proton ions. So flux of proton that happens. So as protons come from intermembrane space to the matrix, because of this flux, energy is produced, and this energy allows for fusion of ADP with an inorganic phosphate group to produce ATP. This ATP can be used up somewhere else, and the reverse process happens. When the reverse process happens, when the ATP divides into ADP and PI, then energy is released if there is a requirement of energy. Okay. ATP synthase is a mushroom shaped multimeric protein complex composed of two proteins, F0 and F1. F0 is the membrane bound portion here, and F1 is in the matrix. Both of these are protein complexes, so they are made up of multiple proteins and not a single unit. These complexes harvest the proton motive force to catalyze phosphorylation reaction involving ADP and phosphate to generate ATP. So the functions of mitochondria are production of ATP, generation of reactive oxygen species in immune cells to kill infectious agents. It can be used to track the family tree because of the presence of mitochondrial DNA, which comes only from the mother side of the family. If you were to track the sequence of the mitochondrial DNA, you get an idea of who belongs to a particular family tree. It also has a role in programmed cell death or apoptosis. Apoptosis usually involves a lot of caspases which are present in the mitochondrial intermembrane space. In an event where the exposure, if the cell is harmed or if it's exposed to a environmental condition which is not suitable for the cell to live, it activates various processes, and these processes disturbs the mitochondrial membrane potential, releases cytochrome C or CYTC. All of these events activate caspases. In normal cell, in a healthy cell, caspases are present but in inactive form. When all of these processes come down, then that caspases are activated, and active caspases can then uh, uh, start a series of events which essentially activates DNA's activity. It degrades the genomic DNA. The cell essentially implodes on itself. It blebs and sort of uh, degenerates. The carcass of the cell can be taken up by other cells if there is a requirement. Okay. Now, ATP synthesis itself is not completely understood, but there is something known as chemiosmotic hypothesis, wherein the F1 unit is composed of 3 proteins. So it's a trimeric protein. This is the F1. So the organization is such that it has 3 sides, one for loose binding, one for tight binding, and one is open. The open surface catches ADP and PI, which then, if required, if there is enough proton motive force, goes into a loose binding state. This loose binding state brings the ADP and PI close together, and then uh, if there is enough proton motive force, the pocket which has ADP and PI changes into a tight binding state. This tight binding state allows for ADP and PI to fuse to form ATP. This ATP can, after the formation, can then move to an open phase and then get released from the F1 complex. And this process keeps repeating to make ATP molecules, multiple ATP molecules. Okay. Next is chloroplast, which is specific to the plant cells. This is mostly found in plants, algae, and some hybrid organisms such as Euglena. Chloroplast is one other organelle which has its own DNA. Like mitochondria had its own DNA, chloroplast also has its own DNA. Chloroplast has an outer membrane, an inner membrane, and the light pigment or chlorophyll is present in the thylakoid membrane. So this is the organization of the chloroplast: outer membrane, inner membrane. Within the inner membrane, there is another layer where the liquidy part of the inside the inner membrane is termed as stroma, whereas the chlorophyll or the light pigment is organized in thylakoids. These are also membranous organizations. The outer membrane itself is porous to small molecules, but proteins or very large molecules can only be transported by translocon by TOC complexes, which is translocon on the outer chloroplast membrane. Movement of material from the outer membrane to the inner membrane happens through TIC, or translocon complex present in the inner chloroplast membrane. And this space in between is filled with an aqueous medium. The inner membrane can then fold into thylakoids. Multiple thylakoids fuse together to form a granum. Okay. Now, the photosynthetic machinery itself is present in the thylakoid surface and thylakoid lumen. The thylakoid membranes are arranged, multiple thylakoid membranes are arranged like stacks of coin to form granum, and the chloroplast itself is the granum itself is connected to itself by a tubule to share the material. The photosynthesis itself happens on the thylakoid membrane because all of the photosynthetic machinery, all of the pigments are present here. Photosynthesis itself is a process of generation of glucose molecules from carbon dioxide and water using solar energy. So this, and one of the byproducts is oxygen. This system happens because of two photosystems, photosystem 1 and photosystem complex 2. These are again multi-protein complexes. Photosystem 2 absorbs the light from solar energy and then breaks down water molecules into protons and oxygen. This creates proton motive force, which can then be utilized with the solar energy to form NADPH from NADH2. These in using photosystem 1. The production of this production, and as well as the hydrogen as well as the proton gradient, leads to production of ATP from ADP and PI using another ATP synthase present in the thylakoid compartment. These can then be used to run Calvin cycle to assimilate environmental CO2 to form sugars. So now we know there are multiple organelles. How are these organelles connected? This whole network is known as vesicular trafficking, wherein a protein might be made at one particular point, but it can transfer to another organelle or within another region in the cell. One second. So the main function of these organelles is to manage the distribution of food, proteins, energy throughout the cell. There are three different organelles that are very important for this: endoplasmic reticulum, Golgi apparatus, and lysosome. All of these organelles work together to maintain the transport chain. Some terms that are important is, if the cell is taking up stuff from outside, then it is known as endocytosis. If it's taking up liquid from outside, then it's known as pinocytosis. If material is being thrown out of the cell from inside, it's known as exocytosis. There is also intravesicular system that delivers protein synthesized in the endoplasmic reticulum to different organelles. We will come to how exactly this happens in a bit. In case of endocytosis, material present outside of the cell binds to the cell surface using some receptor and it gets trapped in a structure known as an endosome. It's a vesicle-like structure. It's a bubble-like structure. These vesicles can then fuse with lysosomes to form latent endosome, and here and so lysosomes have multiple hydrolases and enzymes that can break down anything. So the material that is present in late endosome gets digested because of the material present in the lysosome, and these can then fuse to the Golgi complex and can be used for the secretion. Is there a question? Okay. Okay. Coming to endoplasmic reticulum, this is where most of the protein synthesis happens. The endoplasmic reticulum is of two types within any cell, rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is rough because there is ribosome attached to the surface, which gives it a rough appearance, whereas smooth endoplasmic reticulum does not have ribosomes. And the protein synthesis itself happens on these ribosomes that are present on the RER or rough endoplasmic reticulum. The protein synthesis can be to make integral membrane proteins, proteins for secretion, and proteins destined for different organelles. If there are proteins which are destined for other organelles, they are synthesized with the N signal peptide. This N signal peptide is usually recognized by the signal recognition particle on their target organelles, and that is how the cargo is shifted from the endoplasmic reticulum to the target organelle, which can be Golgi, which can be mitochondria or chloroplast, any of it. So functions of endoplasmic reticulum: synthesis of steroid hormone gonad cells, detoxification, calcium sequestration. This calcium sequestration usually happens in smooth endoplasmic reticulum. Synthesis of protein, phospholipid and carbohydrates. Phospholipid and carbohydrate synthesis is also something that smooth endoplasmic reticulum is responsible for more than rough endoplasmic reticulum. Protein sorting to different organelles, protein modification such as glycosylation. Next is Golgi body, which was first visualized by Camillo Golgi. It's essentially a stack of discs known as cisternae arranged to give three distinct zones. There is cis phase, which receives the material from endoplasmic reticulum. There is medial cisternae, where all of the processing for the proteins happens. And there is trans Golgi network, which creates vesicles and acts as a sorting site of sorts to send out all of the vesicles to their respective positions. So Golgi bodies are responsible for protein sorting, protein modifications which happen in the medial cisternae, and proteolysis if required. So Golgi is not only responsible for sorting where each protein is going, it is also responsible for sorting if the proteins are properly formed or not. If proteins are not formed properly, if there is a mistake somewhere during the formation process, these proteins are no longer useful, and these might be known as misfolded proteins. These misfolded proteins are also sorted by Golgi, and then they are transferred to the lysosome for degradation. Lysosomes are known as suicidal bags due to their role in autophagy, or a process where the cell eats itself. It can eat a part of itself, or it can, during the process of apoptosis, turn inside. Lysosomes might turn inside out and digest everything. If the cells have the ability to eat parts of themselves to meet their energy requirement if there is a starvation. It's extremely acidic. It contains proteases, cytolytic enzymes, and to degrade ingested materials. Lysosomes will fuse with other vesicles, and this fusion allows for degradation of whatever components are present within that other vesicle. It can degrade ingested food material for delivery to vesicular system. It can degrade pathogenic bacteria. It can also degrade old proteins or misfolded proteins. Now, plasma membrane, which is the envelope that covers the whole cell, it is a lipid bilayer. So there are two layers of lipids stacked inside out for to form a bilayer of sorts. So the cell envelope is also a double membrane structure. The cell membrane might have peripheral proteins which are attached only to one, which are inserted into only one of the membranes, or it can have integral proteins like these which span both the membranes. The cell membrane has multiple receptors, transporters for transporting stuff from the outside of the cell to inside. It can have glycoproteins, glycoproteins are modified protein structures with uh, glycosidic linkages present on it, which might the glyco part of the protein usually always faces the outside. So the surface of eukaryotic cells has a layer of glycocalyx. This glycocalyx is responsible for multiple things. It But this is something that you will come to later when you read about proteins and their sorting machinery. Is there a question? Okay. Okay. So there are multiple uh, proteins that are present on the plasma membrane. These might be spanning the whole thing, or only spanning one of the bilayers. Now, functions of plasma membrane: they are responsible for protecting the cells. They are the regulators of what goes inside or what stays outside. They are the sites for multiple receptors, multiple transporters. So all in all, an integral part of the cell. Okay. Now, coming to division of cells. So we know that there is cytosol, nucleus, mitochondria, all of this. Now, all of these, the basic subunits that make up all of this are these four things. Protein, carbohydrates, lipids, DNA and RNA. The genetic material is made up of DNA and RNA. The surface of all the membranes are made up of lipids. Carbohydrates is the energy source, and proteins are responsible for controlling various processes within the cell. Proteins are made up of amino acids, carbohydrates are made up of sugars, lipids are made up of fatty acids, and DNA and RNA are made up of nucleotides. Proteins are responsible for building blocks. They usually have carbon, hydrogen, oxygen, nitrogen, and sulfur within them. Carbohydrates function as energy sources, usually made up of carbon, hydrogen, and oxygen. Lipids also can act as energy sources or as membrane for making membranes. These are made up of carbon, hydrogen, oxygen, phosphorus, and sulfur. DNA and RNA make up the genetic material, can have carbon, hydrogen, oxygen, nitrogen, and phosphorus. Apart from that, there are minerals such as iron, zinc, which are cofactors for various enzymes which will be present within the cell. There are vitamins which are responsible, which are essential for cell growth and nutrition. So when we maintain different cells, we maintain them in a medium of sorts which facilitates their growth. So usually for experiments we use something known as immortalized cell lines. These cell lines can be cancerous or non-cancerous, and mostly for cancer biology labs they use cancerous cells, whereas people who study other functions, they can use cancerous as well as non-cancerous cells. So these cells need media to grow. The media has different components that is essential for the growth of the cell. So DMEM, this is DMEM media. DMEM is Dulbeco's modified Eagles media, which was made initially by Dulbeco. But this is just nomenclature, but what it contains is glucose. It contains various amino acids, lipids, all of that in this mixture. Then the media sometimes needs to be supplemented with certain stuff. In this case, for the DMEM, it needs to be supplemented with sodium bicarbonate. We also provide fetal bovine serum or horse serum to ensure that the the serum has lipids and other components essential for growth of the cell. In certain cases, supplements such as 100x antibiotic, antifungals, all of these are given to ensure that the culture remains contaminant free. Okay. So, if you are giving media to the cell will divide if it is not in quiescent stage. So the cell cycle the cells undergo precise cell cycle that produces two daughter cells. Now the division might be equal, wherein both the daughter cells can then go on to produce more daughter cells, or it might be such that one of the daughter cells exits the cell cycle, whereas the other one stays in that cell cycle. So this event of cell cycle is very tightly regulated, leading to division and duplication. The cell cycle itself can be a mitotic cell cycle or a meiotic cell cycle. Meiotic cell cycle is responsible for production of ovum and sperm, whereas mitotic cell cycle is responsible for various homeostatic processes the uh, like the maintenance of skin, of the intestinal epithelium. All of that uh, is done by the mitotic cell cycle. So there are certain differences between both the processes, but the basics remain the same. So the there are a lot of differences between both these processes, but the major steps remain the same. Meiotic cell cycle has other steps in addition to whatever happens during mitosis. In case of prokaryotic cells, the division happens by just binary fission, wherein the cell keeps on growing and then finally after a certain point it just breaks off in the middle. Whereas in case of eukaryotic cells, it has three phases: interphase, mitotic phase, and cytokinesis. So G1S, G2 phases combined make the interphase. M phase is M phase, or the mitosis phase, is where the sorting of the chromosomes happen, and cytokinesis sort of is a interphase is a phase between my M phase and the interphase. Okay, coming to interphase, it is the preparatory phase of the cell where it makes up all of the components that it will require during the cell cycle. During the G1 phase, which is also known as the growth phase, uh, cellular proteins, enzymes, all of this is synthesized because DNA synthesis, which will happen in S phase, requires a lot of proteins. So all of these proteins are made in the G1 phase. This G1 phase is under the control of P53 gene products. Now P53 is known as the tumor suppressor gene, and it's very essential for cell cycle. If any mutation happens to the P53 gene, then the cell cycle goes haywire. It can either fall apart and the cell dies, or if it can the mutation can be such that there is no control in the cell cycle and the cell just keeps on dividing, giving rise to a tumor. S phase is when all of the before S phase, during G1 phase, all of the growth happens, and what happens in S phase is DNA synthesis. So whatever DNA content is present in the cell just doubles. There is no change in ploidy number, there is no change in the structure of the DNA, but the content doubles. All of this happens very fast to avoid exposure to newly synthesized DNA to mutagens. Next is the G2 phase, where cell will grow, it will synthesize more proteins, so it will make more cellular factors to account for the fact that after the cell division there is a need for double the amount of organelles, double the amount of nutrients, all of that. Between between G1 and S phase, there is a, phase known as G0 phase or a quiescence phase. After G1 phase, quiescent, senescent, and non-proliferative eukaryotic cells can enter the G0 phase. All of these cell types, so non-proliferative cells, for non-proliferative cells, the examples are neurons. Whereas for senescence is a process wherein the cells have reached their limit of how many times they can divide, and beyond this point they can no longer, uh, the internal machinery does not allow them to divide, so they become senescent. So they exit the G1 phase and enter into G0 phase. So it is G0 phase, the cells will enter G0 phase whether they don't no longer can or want to divide. This, in certain cases, they remain there for a long period, such as in case of neurons. In certain cases they differentiate, in which they can no longer go back to the S phase, but this is not a regular cell cycle phase, because it's not, the cells are no longer going to divide. That's all. Okay, now coming to the mitotic phase or M phase. So after G2, the cells will enter M phase, and the DNA condenses. They divide the DNA into two parts, and then cytokinesis happens to precisely divide the DNA contents. The first step in M phase is prophase, where the nuclear membrane is dissolved and chromatin condenses to form chromosomes. It will be a structure like this. So there will be four arms. This is known as a centromere, where centrioles will attach. Depending on the position of centromere, there are different kinds of chromosomes that you will encounter, but that will come in later classes, I think. So the nucleolus in nucleus will disappear. Everything condenses. One centrosome, along which DNA will replicate, will give rise to another pair of centrosomes to coordinate downstream events. So the centriole will attach to the centrosome here, and then there are these poles from which the centrioles give rise to microtubules, which will pull apart all of these chromosomes, making sure that equal amount of chromosome has gone into both the cells. Centrioles are considered to organize the microtubule assembly, but they are not essential. There are experiments where people have deleted centrioles, but even after the deletion, the division has occurred. The cells have divided properly. Metaphase is in this phase. So this was prophase, where the condensation is happening. In metaphase, two centrosomes start pulling the chromosomes using the attached centromeres towards the end of the cell. As a result, the chromosome is aligned along the metaphase plate, which is at the middle of the cell, or the equatorial plane. Since the power of pulling is equal, it eventually arranges all the centrosomes in the metaphase plate like this. And the alignment of the chromosome is very essential to decide the entry of cells into anaphase. If they are not properly arranged, the cell will try to arrange them again. Otherwise, it stops the process of division. Then is anaphase, where proteins attached to each of the chromatids are cleaved, and the sister chromatids are separated as daughter chromosomes. The chromosomes are then lined on the metaphase plates, pulled by the microtubules towards their respective centrosomes. So there is a division here. The chromosomes are getting pulled towards two ends of the structure. The exact mechanism is not known, but there is a very high turnover of the microtubule that happens, a lot of assembly and breakdown of the microtubule that happens, and this might be responsible for providing the force of the movement. So at the end of anaphase, you will see a structure like this. Chromosomes have been prepared for distribution between the sister cells. Next comes telophase, where the daughter chromosomes have moved and attached to the opposite end of the cell. Nuclear membrane starts to form on each of the separated daughter chromosomes and nucleus reappears. Several processes during the prophase are essentially reversed in during the telophase. Next is cytokinesis, or the division of the cell membrane. At the end of telophase, mitosis is over, but the cell division requires distribution of cellular content equally amongst daughter cells. In animal cells, a cleavage furrow forms along the metaphase plates that divides the individual nuclei to two separate cells. So what happens is the uh, there is a furrow like this that forms, and eventually the plasma membrane keeps growing, and eventually both of these fuse, separating out the cells. In case of plant cells, on the other hand, instead of this happening, there is cell plate being formed, and so these are discontinuous pieces of membrane that get arranged along the metaphase plate. Then finally, during cytokinesis, this joins, giving rise to two cells. Okay, so the role of cell cycle: it is responsible for development and growth. It is responsible for development of a single cell zygote into a multicellular system. It's responsible for cell replacement. They Certain cells in the human body have a particular lifespan, like RBCs have a span of 3 months, intestinal epithelium gets renewed every 4 to 7 days. So all of this makes sure that the cell cycle makes sure that the old cells gets replaced by newer cells. Regeneration: cellular damage and injury is an integral part of the living system. Cell division is primary event required for synthesis of lost or damaged organ. Now, in case of humans, not a lot of things can be regenerated, but liver is something that gets regenerated if there is an injury to the liver. If some part of the liver gets damaged, then the other parts make cells to compensate for the function of the lost liver. But other parts like heart, kidney, all of these can't be regenerated. Asexual reproduction, which is common in lower invertebrates, also requires cell cycle. So one individual will essentially divide into two, and both of these individuals will be identical to the mother cell. Both of these daughter cells will be identical to the mother cell. One such example is Hydra. In case of Hydra, when it forms buds, this budding is essentially very rapid cell division happening at a particular point. Okay. The cell cycle is very tightly controlled by cell cycle checkpoints. These cell cycle checkpoints are proteins, which are usually cyclin and cyclin dependent protein kinases. The interplay between cyclins and cyclin dependent protein kinases, or CDKs, make sure that the if everything is okay, if the checklist is complete, only then the cyclins and CDKs allow the cell to progress to the next point. Otherwise, the cell is arrested at that point until everything is all right. The main checkpoints are present in G1S, G2M, and M phase. So one checkpoint is here, one checkpoint is here, and one is in here. P53 gene products are also known to control many of these events in G1S and G2M checkpoints. Dysregulation of cell cycle and control mechanisms can give rise to tumor or the cell dying. In general, any cell after a particular number of divisions enters into G0 phase, where it goes quiescent or senescent and ceases to divide. Whereas in case of tumor, this G0 transition to G0 phase is lost. So the cells lose the control mechanism and they keep on multiplying, giving rise to a cell mass, to an uncontrollable cell mass. These cells then take up nutrients are which are required for the normal cells that are present in the vicinity. Proteins known such as retinoblastoma or RB and P53, both of these are crucial cellular factors responsible for cell cycle control, and any mutations that happen to these proteins uh, give can result in tumor development. Okay, so this is the end of the recap for this week's contents. If there are no questions, I'll go to discussing some of the old assignment questions. Okay. Okay. So, let me just share my screen. Okay. So, the first question is, the structure that is absent in prokaryotic cells but present in eukaryotic cells is ribosome, chromosome, membrane-bound organelles, peptidoglycan layer. Okay. The answer is membrane-bound organelles. Ribosomes are present in both prokaryotic as well as eukaryotic cells. So is chromosome and peptidoglycan layer. The role of bacterial flagellum is in facilitating locomotion and chemotaxis, protecting the cell from osmotic stress, synthesizing proteins, or providing structural support. The answer is facilitating locomotion and chemotaxis. The component of bacterial cell wall that is targeted by penicillin is the capsule, the plasma membrane, lipopolysaccharides, or peptidoglycan. The answer is peptidoglycan, wherein penicillin hampers the synthesis of peptidoglycan. Feature of lysozyme that allows us to protect humans from bacterial infections is that inhibits bacterial DNA replication, disrupts plasma membrane of bacteria, cleaves glycosidic bonds in the peptidoglycan, or prevents bacterial chemotaxis. The answer is it's C. It cleaves the glycosidic bonds in the peptidoglycan. The part of eukaryotic nucleus that regulates exchange of materials with the cytoplasm is the nuclear lamina, the nuclear pore complex, the chromatin, or the nucleolus. The answer is the nuclear pore complex. It allows for stuff prepared inside the cytoplasm to come into the nucleus, or things synthesized, RNA synthesized in the nucleus to go out into the cytoplasm. The role of the mitochondria the role that mitochondria play in programmed cell death or apoptosis is generating ATP for cellular activities, synthesizing ribosome for protein production, producing reactive oxygen species, releasing cytochrome C to trigger caspase activation. The answer is D, releasing cytochrome C to trigger caspase activation. Upon if there is any damage to the cell and the cell undergoes a decision that it will lead to programmed cell death, then the cells release cytochrome C. Then these cytochrome C's go and react with inactive caspases that are present within the mitochondria and convert them into active caspases. Chlorophyll in plants contribute to energy production by converting chemical energy into mechanical energy, storing energy in vacuoles, capturing light energy for photosynthesis, or facilitating glycolysis. The answer is capturing light energy for photosynthesis. The next question is, the plant cells have larger vacuoles compared to animal cells to assist in cell division, to increase the rate of photosynthesis, to store toxic substances and maintain turgor pressure, to provide energy for cell motility. The answer is to store toxic substances and maintain turgor pressure. So plant cells do not have a very well developed lysosomal machinery. So what they do is they have very big vacuoles. These vacuoles are usually filled with liquids, which allows, because the vacuole is very big, it also give some sort of structural stability to the cell, um, and it also helps in storing toxic substances. Function of peroxisomes in eukaryotic cells is breakdown of fatty acids and detoxification of hydrogen peroxide, protein synthesis, energy storage, photosynthesis. The answer is A, breakdown of fatty acids and detoxification of hydrogen peroxide. So, if uh, the cell is producing a lot of reactive oxygen species, this reactive oxygen species can then react with water or other components present in the cell to produce hydrogen peroxide, which is harmful. This hydrogen peroxide can also break down to form uh, reactive oxygen species and other components which are harmful to the cell. So there is a requirement for the removal of this hydrogen peroxide always in the cell. The proteins that are transported from ER to Golgi apparatus through lysosomes, vesicles, diffusion, or nuclear pores. The answer is vesicles. Meiosis is critical for sexual reproduction because it ensures genetic diversity in offsprings, it prevents the fusion of gametes, it doubles the chromosome number in gametes, produces identical copies of parental cells. So meiosis is a process, as I said, responsible for making sperm and ovum, wherein in case of mitosis, the ploidy number does not change. In synthesis phase, the genetic material might double, but the are diploid organisms such as human. So humans are diploid organisms because they have two copies of the same gene. So in case of mitosis, the copy number of any gene remains the same too, but any chromosome remains the same too. But in case of meiosis, there is recombination, there is crossing over, and the mishmash of there is a mishmash of genetic material in layman terms, and because of this, uh, the instead of making two daughter cells, in certain cases there are four daughter cells formed. These four daughter cells can then in certain organisms go on to make four gametes, but in certain organisms these there might be two daughter cells that are of a single ploidy number. Okay, so in case of meiosis the ploidy number changes. In case of mitosis it does not change. That is something that you should know. So answer is it ensures genetic diversity in offspring because of recombination of various alleles. The phase of mitosis during which sister chromatids align at the cell's equator is prophase, anaphase, metaphase, or telophase. The answer is metaphase. The process that is responsible for exchange of genetic material during meiosis is cytokinesis, chromosome condensation, DNA replication, or crossing over. The answer is crossing over or recombination. The decision of to divide occurs as G1 phase, early S phase, late S phase, and or M phase. G1 phase. If the cell has decided to divide, only then it will start making things required for division. Otherwise, it will go into G0 phase. The role spindle fibers play in cell division is it synthesizes ATP for mitosis, it separates chromosomes during anaphase, it creates nuclear envelope, it regulates cell cycle checkpoints. The answer is it separates chromosome during anaphase. That's okay. Are there any questions till now? If there are any questions, please let me know. I'll try to clear them up. Okay, then this is the end of the session. I will be here for a few more minutes. If please ask if there are any doubts from today's session. If there are no more questions, then this is the end of the session. Thank you everyone for attending. Thank you.